MIFOBIO, MIFOBIO, Pat, tu me dis dans le casque. Pat. Tu me dis dans le casque si ça te plaît ma voix. Bienvenue à MIFOBIO, c'est l'avant-dernière soirée.
Hi, Elizabeth. Can you? Là, il y a l'essoufflement des... Ça marche ou pas? Ah. Ça fait rentrer les gens un peu?
Ils mettent juste le son au moment où tu commences.
D'accord, ouais, ils me disent que ça vient de brutaliser la machine, d'accord. Ok, bah, je vous propose qu'on redémarre. Parce que sinon on va être en retard pour l'apéro. Hein. Bah on fait pas d'apéro sinon après c'est comme vous voulez. Donc à nouveau on remercie tous nos partenaires industriels et académiques. Et puis, petite nouvelle du maillot jaune. Donc c'est ce défi d'injection d'un laser dans une fibre. Donc actuellement, nous avons toujours le même maillot jaune. Vous avez encore jusqu'à ce soir et demain, si je ne me gourre pas avec l'équipe qui anime, le Fab Lab, donc le Optic Lab. Donc n'hésitez pas à aller relever ce défi. Alors, à mon avis, on peut faire mieux. On a quand même des bons spécialistes de l'injection optique, donc ce serait dommage d'en rester là. Et alors, pour compléter ce défi maillot jaune, on va vous proposer un autre défi, puisque demain, ce sera la fin de ce tour de France d'injection dans la fibre. Et donc, on va finir évidemment sur les Champs-Élysées par un sprint final. Et donc, ce sprint va être sous la forme d'un challenge la meilleure injection en 3 minutes. Donc pour ça, vous prenez contact, vous vous inscrivez, ou du moins vous arrivez entre 14h et 15h. Et vous aurez 3 minutes pour faire la meilleure injection possible. Et... Ah, donc c'est Thursday, excusez-moi. Vous êtes sûr qu'on est déjà aussi loin? D'ailleurs, j'ai une autre nouvelle, c'est qu'en fait, on va pouvoir prolonger pendant 1 semaine MIFOBIO pour se recaler au niveau horaire. Donc toujours est-il, donc demain, entre 14 et 15 heures, le sprint. Le défi photo, donc là ce sera fini pour 12 heures. Donc si vous voulez vos photos, il faut les envoyer sur ce lien. Bah vas-y, come on.
On les envoie pas sur ce lien du tout, on les envoie sur 2 emails qui sont ensuite Sophie et le mien, mais je vous ai déjà donné vous avez reçu un email il y a quelques jours qui vous donne tout ça. Et il y a plein de photos qui arrivent en ce moment-là. Hein. C'est à jour. Il me reste 2 photos que j'ai pas mis tout à l'heure.
Et demain, c'est pas lundi. Wow. Donc voilà. En revanche, donc de fait, on va bientôt se quitter. Sauf si vous décidez de rester qu'on prolonge. Donc par rapport à ça, donc on a mis en place un... Vas-y si tu veux, Julien.
Alors pour repartir, il y a des navettes qui sont prévues. Donc j'ai tout mis à l'accueil du Bel-en-Bras, dans l'entrée. Donc il y en a une qui part assez tôt à 8h pour l'aéroport. Ensuite il y a 8h30 pour la gare, 2 fois à 10h pour la gare et 13h30 pour la gare aussi. C'est les heures de départ, c'est-à-dire... Il ne vous attendra pas, donc soyez là au moins 15 minutes avant. Et ensuite, j'ai mis aussi un petit panneau parce qu'il y a pas mal de gens qui repartent en voiture demain soir et surtout le 12. Et si vous avez un peu de place et que des gens sont intéressés pour que vous les déposiez à la gare ou à l'aéroport, c'est pas très loin, ça vous fera pas faire un gros détour. Si vous êtes OK, vous mettez votre nom, votre numéro de téléphone, quand est-ce que vous avez prévu de partir, et les gens pourront vous contacter. Voilà.
Merci. Donc si vous avez des questions, vous voyez avec Julien. Ensuite, petite annonce par rapport aux posters, pour en particulier tous les jeunes qui avaient affiché les posters, mais tous ceux qui ont les posters, c'est de les récupérer à partir de ce soir ou demain, pour qu'on démonte les grilles support demain sur le temps du repas, pour libérer l'espace, en particulier pour avoir l'espace disponible le soir pour la soirée. Donc je vous rappelle, il y a une soirée demain soir, jeudi, hein, d'accord, et donc voilà, donc pensez à récupérer vos posters. Et par ailleurs, on a mis en place près du desk, à l'accueil, un panneau avec des offres d'emploi. Donc tous les industriels qui le souhaitent peuvent mettre des offres d'emploi, les académiques. Vous êtes tous invités à indiquer vos offres d'emploi. Et tous ceux qui souhaitent, qui recherchent un emploi ou qui seraient intéressés pour changer de métier et quitter le CNRS, peuvent aller consulter ou trouver un emploi, peuvent aller consulter ce tableau, et ensuite on récupérera les annonces et on va essayer de vous les remettre aussi en lien pour que vous puissiez les retrouver ensuite. Maintenant, une petite annonce concernant la salle de culture.
Voilà, j'espère que la salle de culture vous a bien servi pendant cette semaine, mais maintenant il va falloir tout nettoyer. Donc dès ce soir, il n'y aura plus de culture. On va décontaminer les PSM. Donc à partir de 23h, la pièce de culture ne sera plus accessible. Et donc vous pourrez retirer vos affaires le lendemain. Et on gardera un frigo et un congélateur allumés jusqu'à vendredi midi, pour que vous puissiez récupérer vos produits. Bien.
Merci. Et enfin, notre petite note, on est toujours sur cette stratégie de développement durable et de soutien aux tortues. Et les tortues, en principe, vous remercient pour votre aide. Et donc, il y a toujours l'opération avec la bière, qui a plutôt bien marché la première soirée. Plutôt été un bon succès pour ça. Donc, je ne vais pas vous inviter, mais continuez. Pas forcément à boire, mais à donner pour les tortues. Voilà. Et maintenant, on va passer à la suite du programme, et donc au module, et donc avec Elisabeth en ligne.
Am I up? Merci. Bonjour tout le monde. Ooh, that's not good. Yeah, that was very unnerving having an introduction in French. My mum is actually a French teacher, and I'm going to have her watch the recording of this and have me learn what you just said about me.
Excuse me,
hopefully you can all hear me okay and see the slides.
Excuse me? Elizabeth Hillman? Yeah, the introduction has not been done yet. That was not the introduction. It was just some news about
well, there you go. That goes to show how bad my French is. My mum would be horrified. Okay, I'll try.
So the introduction is coming up. So I'll just start. So Elizabeth Hillman here is a team leader at Columbia University. She has obtained her PhD in medical physics and bioengineering from University College London in the UK, and has then moved to the US for a postdoc. And then, so she did her postdoc in Boston, and then became assistant professor and associate professor at Columbia University, alternating between the departments of biomedical engineering and radiology. And since 2014, she joined the Gcamp Brain Behavior Institute at Columbia University, where she develops novel microscopy developments. And today she will tell us about a new high-speed 3D optical imaging system. Thank you.
Okay, that was much easier to understand. Thank you very much. Okay, so welcome everybody. So I'm going to talk to you about escape microscopy, and I have a conflict of interest disclosure here that the technology is licensed to Leica. Okay, so I know I'm sort of in an organoid session, but what we focused on a little bit more in my lab is all kinds of in vivo imaging. And ever since I began sort of developing imaging systems, I wanted to capture what was going on in living systems. The problem with living systems is that they are three-dimensional and they move. And that is true of organoids as well, but also of these kinds of amazing animal models that have come about over the last few years, thanks to all these advances in transgenic techniques. So we've here got worms and fish and flies. And even things like cellular imaging, things like thinking about mechanobiology and the way that cells move and the way that cells signal and the way they move in response to signals. These are all things that we really would like to study. And I have this little fly walking around here to remind you that all of these little organisms, it's not just that they're three-dimensional, but they move. And so standard microscopy techniques that capture, like in this image, a single plane, are not going to be able to capture information about these different organisms and these even different kinds of cellular functions. And so what you end up having is a lot of expertise that's needed to just actually figure out how to hold your sample still or how to infer what might be happening when the animal is moving if you can't allow it to move. Another huge thing that we've been able to take a lot of advantage of is the advent of things like this, which is a calcium-sensitive fluorescent protein. So what you're seeing in some of these other images is sort of just GFP expression, but there are types of fluorescent indicators now, as well as a lot of different dyes, that can actually give you a report on the function of the cell. So here we're seeing neurons that are flashing in response to increases in intracellular calcium. So again, this is something that it would be ideal to be able to image if we could image really, really quickly. Okay, and also things like optogenetics as well allow us to evoke all sorts of cool things happening in tissues, and we want to be able to capture the dynamic responses to those as well. So probably I heard that this is a microscopy school. I'm so sorry I couldn't be there with you I love these kinds of things and teaching these kinds of things, but hopefully you all learned a lot about point scanning microscopy, confocal microscopy, and two-photon, which is what I'm showing here. So we built the two-photon microscope in my lab, and this is an image of a living mouse's brain, and we were able to scan a little spot of light around, like you see here, into a three-dimensional cube or a block. And here pick up really nice images of astrocytes and blood vessels in the living brain. And we can get down to about 500 microns in depth. The problem is that this whole stack took about 5 minutes to acquire. And that's because when you do two-photon microscopy, in general, if you want to be able to get a really nice crisp image, you have to move that little spot of light around to every single point in that three-dimensional volume, okay? So if we just even pick a more conservative volume, let's say 400 pixels by 400 by 100, that's 16 million points. But what you should have been learning about two-photon is that it typically uses a Ti-sapphire laser that has to be pulsed. And most lasers that you can use for this are pulsing at 80 megahertz, which is 80 million pulses per second. So it's very easy to do this math and say that we wouldn't even be able to capture this volume any faster than 5 volumes per second if we were allowing there to be just one laser pulse per pixel, which is not enough to really give you a lot of signal anyway. But so 5 volumes per second really isn't fast enough to capture all of those dynamics, and this volume here is very small. If you were just doing CW and not using a pulse laser, what you would end up with is an integration time per pixel of just 12.5 nanoseconds, which is absolutely tiny. In fact, it's getting almost close to the fluorescence lifetime of green fluorescent protein. So you can see that it doesn't matter if you just make scanners that are much, much faster or piezos that can bounce your objective lens up and down quicker. You're going to reach this limit of available signal to noise and integration time. And it's going to mean you're going to need to use really, really high powers. And that's going to also cause your sample to die much quicker. Also, as I said, this volume is simply not even enough to capture large areas. And in this case that I've given you here, you would be scanning your beam at 200,000 lines per second, which is about 10 times faster than your average resonance scanner. So we tried to do this. We tried to build the fastest two-photon microscope that we could, but we realized when we did these calculations that there wasn't going to be a way for us to make a faster imaging method if we were only scanning a single spot around. So around the time that I was starting my lab, I started to see these beautiful images from light sheet microscopy. So here, this is ultramicroscopy by Hans-Ulrich Doth. And I remember seeing this and saying, hold on a minute, you can't do microscopy like that. That doesn't make any sense. But yeah, we were seeing these gorgeous, beautiful images that were being formed in a completely different way. And then this work here by Raju Tomar and Philip Haller really emphasized something very special about light sheet microscopy, which is that it doesn't kill your sample. It's here looking at embryogenesis of a Drosophila larva, and you can see that they were able to image here. This is 6 hours coming up to 7 hours, and capture this full three-dimensional evolution of that development. So this goes to show that the technique is very gentle on the tissue compared to needing to use a lot of high power. But, you know, although Lightsheet has these great properties, you'll see that actually nobody was really using it to image anything fast. Although this video has been sped up, we've now actually just watched 10 or 11 hours worth of development. So each of these images took many, well, I think maybe, 15 seconds per time point to acquire. So this wouldn't be suitable for looking at actual, like, living animal dynamics. In fact, at the end here, when it actually starts to move, you can see that the image kind of falls apart and the animal kind of crawls off. Okay, so we wanted to think about how could we leverage these advantages of Lightsheet, and it actually took us a while to kind of realize exactly what they all were, but I'll give you a little bit of a background. So the thing that you'll hear and that I was just explaining is that Lightsheet has low photobleaching, right? And this is a plot actually from Tim Hawley's lab, who has a Lightsheet technique called OCPI. And he did this very nice demonstration that in the same sample, here he could image up to 20,000 OCPI images and still have here about 60% of the fluorescence left. Whereas when he was using confocal, he was down to 20% after just about 200 images, sorry. Okay, so you can see experimentally this holds true. But why? Okay, so the first thing that people will tell you is it's because of this selective plane illumination. So that's what actually some people call a Lightsheet, is SPIM, selective plane illumination microscopy. So that's really good. It basically means that here I'm only putting the light here into the plane that I'm imaging at any given point in time. And we can compare that here to confocal, to spinning disk confocal, and to Lightsheet. So although in confocal what you're doing is you're making a high numerical aperture focal point here, and then you're using a pinhole up here to get rid of all of the light that might be coming from places that aren't that focal point. What you can see is that you're actually putting a lot of light into the tissue above and below your point of interest. And then you're actually just using this pinhole to get rid of it again. But all of that light is going to be causing photo damage as well as basically wasting signal. And the same is going to be true of spinning disk confocal. But in Lightsheet, you can see that we're not actually illuminating those areas above and below while we're actually collecting the signal. And so that has this huge advantage of saving this tissue above and below and not causing it to be imaged again and again or illuminated again and again as you generate a stack. But there's something else as well that actually gives a major advantage, and it comes to what I was saying before about integration time. So when you visit each spot one after the other, then you only have a very, very small amount of time to see each individual one. But if you have a camera that is collecting an image of that entire plane all at once, so a 400 by 400 pixel plane, then your cameras are the camera pixels are going to be detecting signal for 160,000 times longer than if you were point scanning. And so that means that we can use cameras and that we can get actually much higher signal to noise images over that entire plane because we have a lot more time to collect that light. We can also therefore use less light at any moment in time, and that's a lot better for fluorescent proteins in particular that have a sort of nonlinear photobleaching, according to the papers, nonlinear photobleaching for higher powers versus lower powers. And so we can use less power density. So the other really interesting thing is that we're also giving photons multiple chances to interact as they travel along the sheet. So if we follow this little photon here, that's going across, he has a lot of different chances to interact and generate an emission fluorophore, right? And it's shown as well up here. And so the chances of a photon actually exciting fluorescence are relatively low. And so the first pass, you know, you may only get 1% or zero, I don't even know, tiny percentage of interactions that are going to generate fluorescence. But because those photons are all traveling in the same direction and that we're imaging that entire plane, they have many, many, many chances to create fluorescent photons, and we are interested in all of them. We're not blocking them. In confocal and in sort of these wide field techniques, you only want to capture the interactions that are occurring at one single point in time. Okay, so you massively decrease the number of interactions that you're actually able to collect. You don't give them any chance to have meaningful interactions above and below. Okay, and that means you get way more emission photons per milliwatt that you put into your sample compared to these high numerical aperture techniques. And so we can model this, and we have a little paper here that shows these comparisons. But essentially what it says is that it's not just a gentle method. It's not that just that you're not photobleaching, but it's actually a very efficient way of generating photons. And so if we were to want to make a very high-speed three-dimensional imaging technique, what it means is that even on a practical level, if we can make the images, that's great. But this shows that if we are making the images, we can actually maintain the amount of fluorescence in our sample and get good signal to noise. In addition to everything else. Okay, so there's the potential for it to image very, very fast. So what's the problem? So conventional light sheets look like this. So they create a sheet of light in the sample, and then they focus the detection, the camera, onto that light sheet. Okay, so that creates this very nice kind of three-dimensional point spread function here, which is limited by both the numerical aperture of your detection as well as the thickness of your light sheet. But most people who are trying to make three-dimensional images here would either move the sample plane up and down. So in this case, you're scanning the light sheet with one scan arm over here, and then you're physically moving this lens up and down. Or you have to physically move your sample up and down. And none of these things matter if your sample is a piece of dead, clear tissue and you have a lot of time. But if you want to actually image something that's alive, you definitely don't want to shake it up and down. And if you want to have this moving, you're going to be disturbing your sample. And there are techniques where you can kind of do this electronically, but getting them to synchronize between these two different modes of scanning is really challenging. And so typically, these kinds of studies have not managed to get to faster than about 1 volume per second imaging speed. In addition, you can see that these two lenses with this kind of need for access to the sample all the way around means that you have to get used to these rather complicated methods of mounting your sample. And so, for example, if you wanted to image this fish and have him behaving and doing things, it's going to be very difficult if you have to embed him in agar and kind of hover him in this three-dimensional space. And so this is the standard, though, for the kind of commercial light sheet systems that you can get, that you have these two lenses and you have this kind of need in here to somehow translate relatively the sheet and the detection plane or the sample in order to create something that's three-dimensional. And all of that stuff takes time. Okay, so what we do instead is we illuminate the sample from above on an oblique angle, and then we collect the light back through the same objective lens. And so this little diagram here is showing how we actually get our point spread function. So it's the product of the excitation point spread function and the detection point spread function. And so here, even though we may not have an exactly 90-degree angle here, we can still get relatively good isotropic resolution. And so, but what's really nice, if you have this geometry now, is that you can actually get rid of your sample hovering in free space, and you can actually image things like intact samples. So you can also use immersion lenses, and you have a lot more freedom about the numerical aperture of this lens and the working distance, because you're not trying to physically fit all of these things together around your sample. So this turns light sheet into something that's much more equivalent to like confocal and two-photon microscopy or even epifluorescence microscopy. So one thing here that now everyone always gets confused is they tell me that, you know, it's not possible to make an image using an objective lens on an angle like this, that, you know, we should only use a lens where we have a horizontal focal plane. But in fact, it is possible to generate this very cool oblique focal plane so that we can truly get this light sheet imaging condition of our detections focused on this sheet while our sheet is obliquely entering the sample. Okay, and this has been really well described previously by Chris Dunsby here, who described this technique of being able to map and image this oblique plane very nicely. Okay, and so we call this technique swept confocally aligned planar excitation microscopy, because the real magic here is we haven't just created this imaging geometry, but what we need to do now is make a three-dimensional image. Now, we could move this sample backwards and forwards, or we could bounce the objective lens up and down. That would be quite standard. But we found a way instead to move the sheet from side to side so that we can actually sample this three-dimensional volume without anything needing to move at the sample. And I'll show you how we do that now. So here's a basic layout of the system, and it's actually very similar to a confocal microscope. But instead of bringing in a high numerical aperture beam here in the middle, we bring in a low numerical aperture beam off to the side. So when this hits the back of the objective lens here, it creates this oblique sheet coming out down at the sample. And because we've got a cylindrical lens here, this is extending like into the board as the sheet, right? So if we look at what's happening to the light that's coming back, we put it through this other second arm here and we recreate the image that we would have down here, okay? And this is utilizing something called perfect 3D imaging that allows us to actually reproduce the entire three-dimensional field down here at the sample over here in this imaging plane. Now, if we then want to take an image and focus on that plane, all we have to do is take a third objective lens here and focus it onto that plane. And now I'm mapping that plane, that illuminated plane, straight onto the face of my camera. So essentially, the camera thinks that it's down here, and it thinks that it's localized and focused onto that oblique plane. Okay, so that's how we've generated our three-dimensional pattern down here at the sample. But here's the magic part about the scanning. So we have a galvo mirror here. And this works just like in a confocal microscope. So when you move that mirror from side to side, it causes what would be a spot down at the bottom to move from side to side, right? But, oh, hang on. Something crashed. Sorry about that. I'll be back in a minute. Okay. Sorry, my computer's had a hard week. Let me just... Okay, sorry about that. Okay, so as we move that sheet as we scan this back and forth in a confocal microscope, that would be scanning the spot from side to side here, right? And in a confocal microscope, what we would say is that the light now coming back is descanned off this mirror, right? So these rays still hold true. These rays are going to bend this way when the mirror is moved, but when the light's coming back, it's going to be corrected and it's going to follow this same path. So in a confocal microscope, you would put a pinhole here and you would just detect the signal that was coming back and make your image. In our case, this has that same effect. So the sheet moves from side to side at the sample, but the focus on the sheet is maintained even when the sheet is moving because the galvo is descanning that. Okay, so the image always stays aligned with the moving sheet as the sheet moves from side to side because this mirror is correcting that movement. And so this plane over here is always focused on the light sheet. So this achieves that same thing of having a sheet and having your detection plane focused on it and then moving the two together. But both of those things are just done by this one mirror by itself. So wherever this moves, it's always going to maintain the focus on the light sheet down here. And so what it means is we just can move that mirror backwards and forwards and capture this image of this little fish here. And this mirror only needs to move at the volume rate. So if we want to do 20 volumes per second, this mirror just moves at 20 lines per second. So this is not a resonance scanner. This is a very cheap, very simple galvo that's just moving from side to side while we're just streaming images on the camera. And this already shows you an advantage of this geometry where like the little fish can just sit there, maybe in a drop of agar to immobilize it. We're not shining light into its eyes, which it really, really hates. And we're able to capture and sample that full brain there. And so here's just an example of some images. This is dual color, by the way. I'll explain that in a minute. Coming out of the system, they come out much, much faster than is shown here, but they're basically Z and then X. And you can stack those together and turn that immediately into a nice three-dimensional image. So the dual color comes from us putting an image splitter here because this is a simple imaging system. So the image splitter can project the two images side by side on the camera. And that allows us to simultaneously collect multiple colors at the same time and doesn't waste any speed because we're collecting the same number of rows across the camera. Okay, so also you can actually read this camera out really, really fast if you're going to only image, for example, 100 or 200 rows, which is equivalent of having 100 or 200 depths into your sample. You can image, you can use a standard Xyla camera to image at about 2,000 frames per second if you're only capturing 100 rows in depth. And that means that you can use all 2,000 of those frames to form your three-dimensional volume. So there's no reconstruction here. We don't have to deconvolve or anything. This is truly optically sectioned images. There's no translation of the sample in Z at all. So this thing is completely stationary. In fact, the only moving part is this one galvo mirror. I'll just make note. So this rotation part here is the trickiest part of the system. It's quite challenging to actually capture all of the light here into this third objective lens, and that's the fundamental limit on the numerical aperture of the system and thus the resolution. So there's been some really nice work in the last few years that has developed this very cool method of basically making a zero working distance lens, which is embodied here in the snouty lens that's now been developed to do this. And so it has this very, very cool effect that if you actually put a piece of glass right at the focal plane, then this change in the direction of the light as it's entering that higher refractive index medium actually bends all of that light into the third objective lens. So using this approach, you can actually capture all of the light that's coming back from your primary objective lens, almost all of the light, greatly improving the NA and allowing lots of people now to use this approach for much higher resolution sort of cellular and subcellular imaging. Okay, so, but this is our basic system. So this is a system that we brought with us to Dresden a few years ago for a light sheet conference. We put it all in a suitcase and we brought it and we built it there. It's just got a simple 488 laser back here. Here's the galvo mirror. Here's the primary objective lens pointing down to the sample. So again, this is all stationary. This is just a three-dimensional positioning stage to put the sample where you want it. Then the light comes back off the galvo mirror over to here. This is the rotation part. This is the image splitter, and there's the camera. Okay, and one of the really nice things about this sort of isolated objective lens is you can do crazy things like this and basically spin this objective to any angle that you want and be able to do, say, inverted or upright or even off to the side kind of imaging. So this design actually also comes out as being really very inexpensive, very simple, and very robust. Here's an example of data being collected here. So this is the light sheet in the fish. So this is those images, those oblique images of the fish. Down here, you can see the fish is just sitting in a dish under this 20x 1.0 NA water immersion 2mm working distance lens. And yeah, there's the images just flying out, and there's Venkata, who put his heart and soul into developing this system during his PhD. Okay, and again, because it's low photo bleaching, it's just sitting there. The galvo mirror is actually not moving very fast. Everything just kind of is pretty straightforward. We were imaging these fish for an hour while they were having spontaneous epileptic seizures and generating about 1.5 terabytes of data in that hour. Okay, so why do we actually need this fast 3D imaging? I mentioned this a little bit earlier on. So this is an example of a calcium imaging data set where we're using, as I mentioned at the beginning, this calcium sensitive fluorophore called Gcamp, which increases its fluorescence in the presence of an increase in intracellular calcium, which is what happens when neurons fire. And so we captured that epileptic seizure activity throughout that whole three-dimensional brain. And it's not something that you could do if you were building that up from a bunch of different planes that you were collecting at different points in time. So you can do all sorts of nice analysis on this. For example, here, just looking at the time to peak of that wave of the seizure and being able to map back exactly to where in the brain it started. You can also then look at how it actually propagates and how it changes every single time. And the person that was working on this in the lab was very interested in turning this into a sort of high throughput screening technique for looking at anti-epileptic drugs by suppressing these seizures, being able to kind of microfluidically move the fish into the field of view and out and do high throughput screening. I'll give you another example of a recent paper that we did that was using this same technique to take advantage of our ability to image this very large three-dimensional field of view at high speeds. And so this was to look at the olfactory epithelium in the mouse. So this is a pretty a gross image of it's an in vitro perfused alive hemihead preparation. And what we're imaging here is calcium sensitive Gcamp, again, in olfactory sensory neurons. Now, these neurons each express a G protein coupled receptor, which binds to specific odors. And this is how the brain actually gets the information about what you're smelling, because certain subsets of those receptors are triggered by all kinds of different smells. But we were able to do this side view and capture this full three-dimensional shape of these turbinates, which otherwise people would use dissociated cells in a two-dimensional plane, but they would no longer be connected to the brain. This is work in collaboration with Stuart Feinstein and my postdoc Owenzi and his postdoc Lou. So we were able to capture the activity of this tissue over time, and what we did was actually we puffed over different kinds of odors and different kinds of mixtures of odors, and then we were able to just isolate the signal from every single one of these 10,000 or more cells and quantify exactly how each single cell responded to each single different odor that we gave it or different mixtures of odors, and do this like over and over again. And that allowed us to profile then these kind of response patterns across these many thousands of odors. And we found something really interesting, which was that sometimes their response could be modulated by the presence of other odors, but they could either be enhanced or inhibited by the presence of another odor, even if they didn't respond to that odor by themselves. And so this was something that we knew from sort of odor experiments, that if you mix together, say, chocolate and orange, then you may predominantly just smell the chocolate part, right? But we didn't know whether or not that actually happened in the nose or whether it happened up in the brain. And so turned out that was kind of a really cool thing, and we published that in Science last year. Okay, another reason why we need high-speed imaging is because things move around. So I just showed you kind of calcium imaging, but now this is imaging at 100 volumes per second, the beating heart of zebrafish. I hope these movies are coming through okay. I'm not actually checking the chat here. So yeah, this is 100 volumes per second in a beating zebrafish heart, 2 colors. So we're looking at red blood cells there going in and through the heart. And again, this is a fully real-time image. It's not made up from a bunch of different layers or stacks that were triggered by the heart beating. This is just capturing the full 3-dimensional beat as it's going on, and that allows us to do quantitative analysis like you saw here of tracking individual red blood cells, seeing how often they actually stay, and one of them gets stuck and kind of stays in there for a long time before he makes it out, which is kind of cool. We can also do calcium imaging in this prep. So this green now here is actually that Gcamp again. So we created a deep learning model to track the 3-dimensional location of the cells and then extract the calcium information so that we could start to look at the way in which the calcium dynamics change as the heart is beating. And we have a red color marker there as well, so we can ratio out from calcium. Okay, in another study that I did with Wes Gruber and that was led by Rebecca and also Wenzi in my lab, was to apply this to the development of fruit flies. And so Wes was super interested in these proprioceptive neurons, which are neurons along the fly wall that are supposed to be reporting back to the central nervous system the position of the body of the larva. But Rebecca was trying to image them and found that, she had to actually immobilize them and stop them from moving so that she could image them using her spinning disk confocal. But then she couldn't say that they were detecting movement because the animal wasn't able to move. So we set this system up where we just literally stuck them on our system and let them crawl. And this is imaging at 10 volumes per second. This is GFP expression in these proprioceptive neurons on the dorsal surface of the animal. The top image is showing a maximum intensity projection from the top, and the bottom is showing the same thing from the side and at the same time. Right? And so we can see this really cool kind of crunching and crushing of the dendrites of those neurons as it crawls. And then this is the image from below, the ventral side. And again, we can see these proprioceptive neurons here actually kind of flipping up inside of the body, like folding inside of the body as it goes along. And this thing here is the ventral nerve cord, is actually the central nervous system of the fly larva. And I'm showing these data sets where they're actually full 3-dimensional volumes over time. So you can pause them at any time and you can do cool things like this and actually track exactly the position of each of these processes and so on. But the really nice thing comes when you actually add the 2 colors. So now we can do a static marker, which is TD tomato, plus this active marker, again, the Gcamp. So we can look at now not just how they get deformed, but how they fire as they're getting deformed. And so you can see these really nice patterns of the dendrites getting bright, and then this little axon thing here as well, which is showing the activity pattern of the neuron as it's being moved and squished as the larva is crawling along. Now, analyzing this kind of data isn't terribly easy. We had to develop tracking algorithms that would actually track the cell body here as it was moving. But that was nice because this gives us the actual behavior. So we have the acceleration of the cell, the distance between the cells and everything like that. And then we can extract the Gcamp signal from the location that we're actually tracking. And we did dual color labeling here again with green and red, so that we can ratio and actually find then both the movement pattern, which is the dotted line, and the firing pattern, which is the Gcamp signal. And so we did this actually for a range of different proprioceptive neurons. So it turned out there were like a lot of different ones that have all these different morphologies and like anchor points. And so instead of just sort of doing one, we did it for this whole different group. And we found some really interesting things about the way in which they each fire at different times. So when people have looked at these before, they said that, okay, fine. So they fire when the lava's body, moves, like who cares? And if you ablate them, you can ablate a certain kind, you end up with the lava just moving slowly. So they were like, there's redundancy in this system. But when we put all of these together, we found that actually there was this very nice kind of complementary pattern of activity between these different cell types. And you actually can see from their morphology that they're positioned in exactly the right place to be either stretched or compressed during each different kind of type of movement that the animal was making. So we were actually able to turn this into a model where now the lava could pretty much do anything it wanted, and we could predict what it was doing based on the signals that we were detecting in these different neurons. Okay, this is just a complete aside. This is not biology at all, but we actually developed this very cool collaboration with a volcanologist who was very, very interested to learn the way in which liquids and bubbles and solids interact with each other in 3 dimensions in order to develop mathematical models that could explain why volcanoes sometimes just bubble and why they sometimes explode. So Julie came into the lab and worked with Creeper in my lab, and we developed these very weird little kind of phantoms and all these new ways to kind of track and look at these different 3-dimensional patterns here, looking at the growth of a bubble at very high speeds and being able to analyze exactly how it nucleates, how it grows, and how it rises off, afterwards. And so this was published in Geochemistry, Geophysics, and Geosystems this year. Okay, so finally, I've shown you that things are fast, things fire. I've shown you that things move. The other reason that we kind of like imaging fast is because imaging is very boring when it's slow. I was really sad to see this stock photo of microscope boredom. Maybe some of you have experienced it. I haven't really used that many commercial microscopes myself, so I'm always surprised to find the settings and say, wow, why are we going so slowly here? This is a piece of expanded mouse brain tissue from Ed Boyden's lab. And when you expand the tissues, you get this gorgeous resolution. But this was a 5 millimeter long strip. And on our system, we were able to image that whole strip in just 8 seconds, just whizzing across, as opposed to it taking a very, very long time if we were doing it through confocal stitching. And I have this actually on the next slide. So by putting this together, we found now to be able to image the tissues very, very quickly. So this is a cleared mouse brain where we actually collected the data as kind of strips going across, and then Creeper kind of stitched them back together. But we get this really, really nice resolutions, really isotropic, reasonably isotropic sampling throughout the data. And this is a 400 micron thick slab, was imaged in about 230 seconds. So we've actually just got a grant to extend this to try to build a system that's going to be able to image the entire human brain at cellular resolution. And again, this is the resolution that we get in individual pieces. And I showed this flat mount retina because I met somebody at a meeting one time who came to me afterwards and said, I spent all my days sitting on the confocal train to image these retinas. And so he sent us some. This is not clear, this is just fixed. And we were able to, again, image this whole thing in about 127 seconds. So just about 2 minutes, just by going kind of across, generating these strips and then putting it back together. So there's a lot of things that we can do here. Here's zooming in on a little part of it, showing that really brilliant resolution if we go to a higher magnification. And this was all featured in the paper that talked about this work in 2019. I threw this in here as well because we've got some organoids. So we have a collaboration with Dominique Paquet in Munich. And so this also happened at the Dresden conference we went to. There were people that were working in these in vitro systems with microfluidics and so on, really, really wanted to take advantage of the properties of light sheet, but could not get these things to fit into any kind of like conventional light sheet geometry. And so we show here that we can actually very easily image within these chips with no problem, just actually intact. And this opens up a lot of really, really cool things that we can potentially do, not just to capture, how these things are growing over time, but to think about capturing things like dynamics of transcytosis, having two layers and looking at how things move in between those layers, also thinking about looking at how things flow and how there may be calcium signaling and things like that, all nice and gentle inside of your chip. And as your chip is growing over time, you can just set the whole thing up to work for a very long time and generate a lot of data. Okay. And I'll just put this in here. This is a new paper that's hopefully going to be coming out in the next month or so, I hope. This is work from Creeper in my lab, and she basically took the system and decided that she wanted to see if we could use it for clinical imaging. So she got lots of fresh mouse tissues and just sort of stuck them under the microscope and found this really gorgeous, all of these images except this one are intrinsic fluorescence. This is just a high-speed imaging of an intravascular dye in a mouse brain where we're just actually moving the mouse around and scanning around in real time. So she developed this method, and this is imaging now a living mouse's kidney and scanning across the kidney and just visualizing the data that's coming out, just as two different planes and a side projection. And then on the left, as she's scanning across, she's actually stitching the volume together. And so we're very, very excited about this for clinical applications to get basically histology type measurements in vivo in living people. And I don't have it here, but we actually imaged the tongue of a volunteer and got some gorgeous images. So we think this could be used for looking at surgical margins, biopsy site identification, sort of laparoscopic tissue typing, looking at reperfusion when you've done a surgery where you've add back into the blood flow, and also looking at transplant organ assessment. And so here's some data on a human kidney where we get this gorgeous imaging just purely from autofluorescence, not from adding any dyes, because of that super duper efficiency of light sheet, but also here giving us really true three-dimensional images that the doctors can explore instead of doing conventional histology. And we're developing a miniaturized version here that will hopefully be able to take into the clinic. Okay, I probably don't have a lot of time because we started reasonably late, but last thing I wanted to just talk about was in vivo mouse brain imaging, because that's where we started. I didn't actually think in the beginning that this system, this escape system, was going to be all that useful for the thing we were trying to do in the beginning, which was high-speed imaging of the mouse brain. But we knew we didn't have any other methods here that could do a good job. Okay, so we here collected data. This is a single photon excitation, so 488 nanometers excitation, capturing a three-dimensional volume of the mouse brain at 10 volumes per second. So there's a red dye in the vasculature now, and the green dye, the green is Tie2 GFP expression in endothelial cells. And so during this real-time data set, so this is exactly what's happening in real time, we're flicking the animal's whisker, and this is the whisker barrel, and so we're able to see this great dilation of these blood vessels, which is another research area of mine to look at vascular activity in the brain. So we can now pick out in this three-dimensional volume this amazing dynamics of the different vessels dilating that you would just not be able to do at all if you were trying to explore this in 2 dimensions. So actually, people got very excited about this. We now have a grant to look at tumor physiology in collaboration with a group that do radiation therapy on tumors. So we've actually installed a system at this beamline, and this is Grace and Salle when we went up there to learn about what they were doing and prepare to install this system onto their beamline. But we can, of course, measure neuronal activity as well. So this is Gcamp activity in dendrites, apical dendrites of layer 5 neurons, again in the whisker barrel. And this was collected at, again, 10 volumes per second, just showing like a top-down and a side view projection of the data. And again, back to the sort of calculations we did in the beginning, this is capturing data at 150 megahertz pixel rate. So that's almost twice as fast as anything you could do on a 2-photon microscope. And we're also using just simple, cheap 1-photon excitation here instead of 2-photon, and still being able to image down about 200 or 250 microns into the scattering mouse brain. So Nick in my lab spent a lot of time working on what we call dimensionality reduction techniques for this data set. So where you basically go through the data, you find the time courses that are common in the data set, and then you do a non-negative least squares fit that finds then how much of each of these time courses is present in each pixel. And then he color codes each of the pixels according to how much of each of the time courses are present, and puts the movie back together. And now we can watch that movie in 3 dimensions, showing really these different dendritic contributions all firing and finding each other. So we call this spatiotemporal unmixing. And it's an incredibly powerful technique that you can harness when you have data that extends in the time dimension in a way where you can utilize that information in these kind of very cool ways. We've built a 2-photon version as well of our 1-photon version to push that depth of imaging further, to get rid of some of this backscattered light, and also to be able to form a focus deeper into the tissue. We weren't entirely sure if this was going to work because obviously we still have some scattering of the light coming back out, but we've got some very nice data here that shows that, oops, it's not playing, sorry. Shows that that we can capture data down to actually about 400 microns. And one of the things we were able to do here is apply that same spatiotemporal mixing technique to be able to pick out not just dendrites, but here now the cell body, kind of the axons around the cell body, we think, and these dendrites, and find different time courses for them within the 3-dimensional volume. And do this, throughout the volume and really map out the presence of all of these different cells that are all projecting to each other and having all these different dynamic signals that you would normally throw away. But now we can actually analyze the way that these different components and compartments of the cell are firing in relation to each other. So that's kind of cool. So there's so much more to say. I didn't talk about fish. I didn't talk about worms. I didn't talk about flies. We've got tardigrades. We've got so many different applications here. And we actually love sharing this technique with people and having them try new things. But I wanted to just give you kind of a taste and an overview. We've been trying to help a lot of people to learn about the system and to use it. I wish I could have been there with you. When we first started talking, I was going to bring a system and it was all going to be super exciting. So we brought systems to Cold Spring Harbor over the years in the trunk of my car. We just bought a new car and we had to make sure it was big enough to let the escape systems fit in the back. And Salle here in my lab has spent a great deal of time documenting this. And so if you're interested, but feel free to reach out. And as I mentioned, it's also under commercial development by Leica. With that, I just want to acknowledge all the people in my lab. So this is my escape team. These are the people that have done a lot of the work that I showed you today. And some the legacy work. So Venkadir and Matt was the very first student to work on this in my lab. And then also, I didn't even really mention it, but I have a whole other half of my lab that looks at brain physiology and blood flow. This is wide field imaging of neural activity all the way across the mouse brain. And having all of these guys sort of working together in parallel is really cool. And these are my alumni from that part of my lab as well. So with that, I just want to acknowledge my funding and thank you for listening. And I hope I didn't talk too fast.
Thank you very much for that really inspiring talk. We definitely have time for a couple of questions.
Thank you for the talk. I had a question related to the autofluorescence imaging that you showed. I was surprised that you could get some signal and still go to 10 volumes per second. Can you hear me? Okay. I had a question related to the autofluorescence imaging. Can you hear me? We can't hear you anymore.
It's hard to hear. The sound is breaking up. Maybe could someone type the question into the chat?
Can you hear me now?
There's a question about autofluorescence imaging.
Okay, let's try again. I was surprised that you could go still at 10 volumes per second and get significant amount of signal. Was that a surprise to you? And was this specific to only a specific kind of tissue? Or because you were showing a lot of examples?
Was it surprising to you? Oh, did you get the question this time?
So I think you're just saying you're surprised that it works.
Kind of.
So I mean, it's funny when I give this talk, I know the first part seems so boring and everyone gets so excited at the end with all the pictures. I should show more pictures at the beginning. But I spent a lot of time at the beginning trying to explain it. It took us a long time to explain it. We were so naive about why it seemed to work as well as it did. So we developed a lot of those models and sort of explanations for why it was working so well, based on the fact that we were able to see what we were able to see. So I don't really know sort of how better to explain it. I think, I'm a physicist by training, and I tend to design these things in a way where I want them to be really efficient. Like, I hate wasting photons, you know, I hate using too much power and then wasting the light that comes out. And so we just designed everything in a way to just keep as much as we could, which led to some trade-offs, right? But a lot of other types of microscopes just don't do that. They, for example, like, everybody's so obsessed with resolution, right, and numerical aperture. And so oftentimes they'll totally sacrifice the health of your sample for the sake of like a super duper crisp image. But if you want to capture neurons firing, you don't necessarily need a really crisp image. You need a really good signal to noise image with a nice healthy sample. So there's just different trade-offs that you can make, I guess. Does that answer the question? It was hard to hear.
I somehow, but so it was really related to the intrinsic fluorescence images that you show. And
the intrinsic fluorescence.
Yeah.
Yes, same thing.
This was very surprising. So
it was a shock. It was a shock to us, but it's true.
And did you see any specificity? Like, do you know if there are some kind of molecules within the tissue that are responsible for this fluorescence? And like, did you study this a little bit more in detail?
Right. So historically, we've actually done quite a lot of work on that. We did some what we call hyperspectral two-photon over the years to really try to tease apart the presence of those different intrinsically fluorescent molecules. We didn't do that here. We were going for something reasonably simple. So we just used 488 excitation and collect green and red emission, basically. And that was enough to let us really differentiate between different kinds of tissues and see structures that allowed us to see, sort of histologically relevant things. We also can use dyes, and we used a bunch of different dyes that are, reasonably well known and approved, like fluorescent and methylene blue and things like that. And those make everything pop as well. And so there's ways we show in the paper that we can do kind of like a pseudo H&E reasonably easily as well with this kind of data. But yeah, it's the same thing. We have people somebody told me it's not possible to do confocal microscopy even in 2D of intrinsic fluorescence. So when we showed them that we did it fast in 3D, they were like very concerned. But we did we did lots of different tissues, and it looked.
Thanks. It's really nice. Thanks.
Maybe one more question, or we don't know. Yeah, one more question if somebody.
I actually have another question, if I may. You just said that you were actually sure that there are some problems where you don't need a very crisp resolution. Did you try to go even further and like design some sort of scape, but in the near infrared or even the second window, and to go at larger depth and image vessels or stuff like this? So I know there aren't so many fluorescent labels that are available at this wavelength, but I was curious about it.
We call it nearscape or near escape, which is a good joke in English. So we've done a little of that, and again, imaging even just at like 800 with ICG, we can get down to easily to 400 microns. We also have a technique, I didn't show it, we call mesoscape. So that actually has a field of view that's about 4 millimeters. And you can leverage, again, this just very, very high bandwidth detection. So, up to 1.3 gigapixels per second. And just spread that out over a larger volume. So we've got some gorgeous images of like whole zebrafish, lava, or we're just doing hydra today, which is super fun to watch. We've kind of done a bunch of different extensions of the technique.
Thanks.
Soon.
Thank you.
Okay.
Thank you very much. I think
By the way, we're hiring, so if anybody's interested, send me an email. And again, so sorry that I couldn't be there with you all, but hopefully sometime in the future.
Thank you very much.
Okay. Thank you.
So our next speaker is Nicola Monk. He did his PhD at the Institut Curie in Paris and then moved to Columbia University as our previous speaker to do his postdoc and is now team leader at the Jacques Monod Institute and his lab studies morphogenesis, and he will now tell us about mechanical sensation.
Okay, can you hear me? Yes? So thank you for the introduction, and of course I'd like to start by thanking the organizers for inviting me and give me the opportunity to share some recent results from the lab. It's actually the first time I'm coming to MIFOBIO, and I'm sure that I will be coming in the following years. Okay, so since I'm the last speaker before the beer hour, I decided to talk about yeast tonight. And actually what I would like to talk about today is a basic morphological behavior which we call tip growth. And tip growth is a morphological process that is very important and that is very conserved among walled cells, so cells that have a cell wall. Okay, and you can find that, for instance, in different type of walled cells, such as bacteria, yeast, fungi, and even specialized plant cells, such as pollen tubes or root hairs, for instance. And for these cells that are non-motile, so these are non-migrating cells, okay, this is actually the sole mean that they have to kind of grow, colonize space, and cover very large surfaces. And I hope you can appreciate that by just looking at the spreading of this fungal colony, where you can see that tip growth and hyphal branching actually allows the fungus to cover very large surfaces, okay? But they also use that, for instance, to mate and to reproduce. So this is tip growth is one way for the two partners to grow towards each other and undergo sexual reproduction. And so they can fuse actually using this kind of processes. And this is also very important for force production, and this is important for different fungal pathogens, for instance, that can exert very large forces in order to penetrate tissues of humans and also plant cuticles, for instance. And I would say that one aspect that we are really fascinated with the process of tip growth is that there is, of course, a wide range of directionality and kind of polarized behavior that you can appreciate just from looking at the different movies in this slide. But there is also a very large range of kind of tip size. And so you can find fungal hyphae that can be as small as 200 nanometer. And so some fungal pathogen actually of plants use that to target and to find some very, very small spaces within the plant cuticles. And you can also, on the other side, have very large hyphae that can go up to 10 or 20 micron. And there is also a large spread of kind of the speeds at which these cells can grow. And so you have relatively slow yeast cells that grow at about a few micron per hour, up to some extremely fast fungal hyphae that can grow at several micron per minute. And these are thought to be like kind of the fastest growing cells among eukaryotes. Okay, and in spite of this kind of large diversity of behavior, size, and kind of speeds, there is one aspect that is conserved among all these cells, is that tip growth is actually, in a way, very well connected to the mechanics of the cell wall. And so these are all walled cells, so they are kind of encased into a layer of sugars, which is called the cell wall. And the cell wall has some kind of analogy with the actin cortex, but with several differences. First, the cell wall is in a way secreted outside the plasma membrane, so it's really encasing the cell, and it's made of polysaccharides, so sugars, and also proteins. Okay, and it performs some very important mechanical function in the way that it defines cell shape at each time point. And so if you want to understand how cells are growing at their tip, for instance, you really have to understand how the cell wall itself is able to deform and grow. Okay, and there are, of course, this process has been studied in many different systems, in pollen tubes, for instance, in yeast and in fungi. And I would say, of course, the detail will always vary, but there are at least like 2 relatively well-accepted principles for how cells can grow at their tip in a relatively fast manner. And so, the first aspect is that you need to of course, fabricate new cell walls. And so for this, there are polarity pathways and polarized trafficking and secretion that will target specific set of cell wall remodeling enzyme to the exact tip of the cell. And this will allow to synthesize fresh cell wall in a way. And this cell wall will tend to be probably more fluid or more plastic than the rest of the cell wall, which is more elastic and define cell shape. And this cell wall will be able to be deformed and to progress in space. And the deformation actually is provided by very large forces that are exerted by internal turgor pressure. So probably you know that plant cells and fungal cells are highly turgid cells. And people have measured the internal turgor pressure, for instance, in yeast, and you can find that pressure will reach about a megapascal. And so this is huge. And just to give you a sense, this is several times the pressure that you find in the bottle of champagne. Okay, and so people have also measured the kind of the thickness or the material properties of the cell wall. And so typically you have cell walls that will range from a few tens of nanometer up to several hundreds of nanometer. And the bulk elastic modulus of the cell wall is kind of analogous to what the elasticity of rubber. So typically 10 to 100 megapascal. Okay, and so then, at cell tips, you make a cell wall that is a little bit more fluid or more plastic, so it can be deformed by very large stress exerted by turgor pressure. And so this is how the cell, in a way, is progressing and grows. But you have to think about the process that pressure is exerting some very extremely large stress actually on the cell wall. So it puts the cell wall under tension, and you can estimate this tension with just simple calculation back on the envelope, and you will find that this tension will reach up to tens of Newton per meter. And just to give you a sense, this is 1,000 times the tension that you need to lyse the plasma membrane. Okay, so this shows you that the cell wall is, of course, vital for this cell. So if you digest or if you have a little hole in the cell wall, then the cell will just explode and lyse and die. Okay? And so the process of tip growth is actually a process very dangerous, especially if you think about this very rapidly remodeling cell wall. You have to think about ways for how the cell wall is able to maintain some kind of homeostasis in terms of mechanics. And so these are kind of the questions that I will discuss today and that are fascinating our lab these days. And so the first one is, of course, a very simple question, which is how do cells actually remodel kind of the cell wall mechanics in order to grow at cell tips? And at the same time, how do they ensure that the cell wall integrity will be intact in order to, while allowing cell wall deformation? And so, for instance, are there some existence of homeostatic or mechanical feedback that will allow the cell wall to sense whether it's strained too fast or too much in order to trigger some adaptive response and allow the cell wall to reinforce and to maintain integrity? Okay, and so to study this process, we are using a model system, mostly a model system, which is called fish and yeast, Schizosaccharomyces. So this is a very well-established system to study polarized tip growth. So you see that these cells throughout the cell cycle, actually they grow at cell tips and then they divide in the middle. And so they exhibit, of course, this process of tip growth, which allows them to maintain this rod shape. And so this is regulated by multiple layers. And probably most downstream, you have the recruitment of very important polarity factors, such as CDC42, that will trigger the nucleation of actin structures. And so you have some actin patches, which are very important for endocytosis. And you have very long actin cables that serve as some kind of highways for secretory vesicle trafficking toward the cell tips. And these vesicles carry to the exact cell tips a lot of material that will allow the cell actually to grow. And in part of this material are these cell wall enzymes that you see here localized at cell tips. So here we are looking at a beta-glucan synthase that will fabricate some part of the cell wall. Is that okay with the sound? It's better like that, sorry. Okay, and you can actually see the dynamic localization of this beta-glucan synthase here at cell tips. And so then the cell wall is being built over time and deformed by very high turgor pressure, and this is how these cells are growing and kind of define their rod shape over time. And so, of course, the great advantage to work with yeast is that we have a bunch of genetic tools. And so, for instance, we have many morphogenetic mutants or many mutants by which we can study the process of cell wall assembly. And so I just gave you a couple of images of classical mutants. So these are like kind of the classical cell cycle mutants that were discovered by Paul Nurse and that can grow very, very long because they just cannot do mitosis. These are mutants in polarity that grow in some kind of round shape because they are defective in activating CDC42. And you have, for instance, here mutants in a formin that nucleates these actin cables, and you see that they have this kind of lemon shape, so they have problem to define their shape. So this is really a great genetic system, a genetic model to study some of the basic morphogenesis process. Okay, and so really the question that I want to discuss today is Syllable Dynamics. And so if you think about the problem, actually one big frustration in the field is that before we started this project, actually the only way we had to look at the cell wall and that people were looking at the cell wall is electron microscopy. And so of course, this precludes any kind of dynamic information. So there is a lot of study where people have looked using transmission or scanning electron microscopy of detailed features of the cell wall, their thickness, their layers, etc., etc. But so far there is no, although we have a sense that the cell wall is continuously remodeling and growing and changing shape, there is no methods to allow to monitor some kind of dynamic aspects of cell wall remodeling. Okay, and so inspired by work that were developed actually by Eva Paluch and also in the lab of Dan Fletcher, which is based on a very simple super resolution in 2 colors, we actually decided with Valeria, so this was a former PhD student, to develop a super resolution method or a sub-resolution method to compute a cell wall thickness all around living and dividing cells. And the idea is pretty simple. Actually, we tag the inner face of the cell wall with 1 fluorophore. So here we are labeling the inner face of the plasma membrane, basically, using an integral membrane protein tagged with a GFP at the cytoplasm tail. So you see that it will decorate, basically, in green the inner face of the cell wall. And then we add in the medium some lectins. So lectins are glycoproteins, and they will recognize some specific sugars that are secreted specifically outside on the outer face of the cell wall. And we can, of course, purchase this lectin with different fluorophores. And so you add you see that you have a second layer that basically decorates the outer part of the cell wall. And the rest is very simple, and I'm sure that for this kind of audience, this is mostly trivial. So if you trace a line orthogonal to the surface, you will have 2 Gaussian peaks, and you can, of course, detect the center of these peaks with a relatively high precision, typically 10 to 20 nanometer. And the distance between the 2 peaks corresponds to basically the local thickness of the cell wall. Now the innovation here is that we can actually, because the signals are very bright and stable, we can perform that all around living cells and in time lapse. And so for instance, here we are just propagating these perpendicular lines, and we have a whole plot of the contour of the thickness of the cell wall around a living cell. And we like to actually represent this kind of thickness with some kind of color maps, where I hope you can already appreciate that there are very interesting spatial patterns in how the cell wall thickness is regulated around cells. And so here, these thick parts of the cell wall, which are about 250 to 300 nanometer, they correspond to what we call the birth scars. So the birth scars mark the previous sites of cell division in yeast. And I hope you can see that the cell tips, which is the growing part, actually is a little bit thinner than the rest of the cell. Okay, and so of course the goal is to perform time lapse of that. And so for the first time, actually, we've been able to visualize at least one important mechanical parameters over time in this time lapse, and we can actually monitor kind of how the thickness is evolving over time, and this is kind of a balance between cell wall assembly and cell wall deformation. Okay, and so as I mentioned in the first picture, there is one first striking aspect that we observed, and it was systematic, at least in fish and yeast, and recently we found in other fungi that it's not always the case, is that cell tips are always much thinner than the sites where cell growth is targeted is always, as a cell wall that is always a bit thinner than the rest of the cell, okay? And you can see that in normal cells. I hope you can appreciate that you have this kind of blue part at the cell tips, but also you can kind of manipulate or explore different ways by which these cells will polarize. So when they germinate out of spores, they form this single growing tip, or when they grow out from a spheroplast, which is a cell that lacks a cell wall and rebuild the cell wall, they break symmetry and they form a single growing tip. And you see that this growing tip is basically thinner than the rest. And here this is the little shmoo. So this is again the mating tip that they will use to mate and find their partners. And you see that this site of cell growth is a little bit thinner than the rest of the cell wall. Okay, but the most interesting aspect that we found is by just performing time lapse. Like actually there is, a cell wall thickness that is smaller or thinner at cell tips as compared to the rest of the cell, but there is not a defined thickness. In a way, this thickness appears to fluctuate a lot or to oscillate during the cell cycle. And so, for instance, here these are individual traces of movies that we've been doing of the cell wall, where we're monitoring the thickness here on cell side. So each color corresponds to one individual cell. So these are typically time lapse that are performed over 60 minutes with a resolution of 6 minutes. And you see that on cell sides, the cell wall thickness is relatively stable. But if you look at the same cell at cell tips, you see that you have phases where the cell wall becomes thicker, and then it becomes thinner, and then it becomes thicker again. And so it's not really some kind of oscillation, but there are some fluctuation with some kind of feedback, which allows to maintain the values of cell wall thickness at cell tip around a kind of a mean value that is probably compatible with cell survival. Okay, and so one way to actually replot this kind of fluctuation with feedback is to use what we call a nomiostatic plot. So here what we are doing is that we're computing the cell wall thickness at cell tip at the beginning of a movie. And then in the next 20 minutes, we will test whether the cell wall has been thinning or thickening by just measuring the slope or the evolution of the thickness over time. Okay? So if you start with a cell wall that is a little bit thicker than normal, then you see that the slope is negative, so it means that in the next 20 minutes it's been thinning, okay? And if you start with a cell wall that is a little bit thinner, actually, you see that the slope is positive, so it's been thickening over the next 20 minutes. And so because this slope is actually negative, this tells you that there is a homeostatic system that basically maintains cell wall thickness at cell tip around mean value that are probably compatible with survival. And the intersection of the slope actually with the x-axis gives you the mean value of the cell wall thickness at cell tip. Okay, and so starting with this kind of analysis, we decided, of course, to build on the power of yeast genetics and to perform a genetic screen. And so one pathway that we targeted actually is the cell wall integrity pathway. So this is something that has been studied for many years in the yeast and fungal system. So this is a pathway that is known actually to correct for errors in the cell walls. And so it's been mostly studied by looking at how when you treat the wall cells with, for instance, chemical that will digest the cell wall, how the pathway will become activated. And so there are very interesting features about the cell wall. The first thing is that, about this pathway, sorry. The first thing is that it starts actually very upstream with some kind of transmembrane sensors that are directly embedded within the cell wall. And so I will talk actually in the second part of my talk about these sensors. And so these sensors, we don't really know how it works, but they are probably able to detect some kind of problem into the cell wall. And then they transduce a cascade downstream that will activate a very important raw GTPase called RAW1 that will trigger cell wall synthesis, and also the activation of a transcriptional response through a MAP kinase called PMK1 that will activate a set of genes that regulate cell wall synthesis. Okay, so this is a homeostatic pathway that, in a way, was thought or has been known to promote survival and cell wall integrity for many years. And what we found actually is that so basically we screened all mutants in genes of this pathway, and we found that there were two mutants that were kind of defective in this homeostatic system. So a mutant in a gene called WISC1, which is one of these sensors actually, and a mutant in a ROGEF called RGF1, which is a very important activator of RAW1 for cell wall synthesis. And in this mutant, what you can see is that the homeostasis is kind of defective, so meaning that when you start with a cell wall that is a little bit thinner, then you kind of don't realize that. The cell does not realize that the cell wall is too thin or too thick, and it keeps on doing its own thing. And so this has one very important consequence, is that, and this was known for a long time in the field, that these mutants are actually losing many of their cells during the cell cycle. So typically in these mutants, you have 10 to 20% of the cells that are dying at each cell cycle. And for instance, the most penetrant phenotype is RGF1. And so during the cell cycle, these cells will grow, and then they will, the cell wall will have a problem, and they will lyse just by piercing their cell wall. And so of course, now that we have the method to image cell wall dynamics, we can look at this kind of lysis events. And so typically what we see is that you start from a cell wall that can be a bit thick, and there is not much growth in these mutants. So here you are using this RGF1 mutant. And very rapidly, you have these phases where the cell wall becomes thinner and thinner and thinner again, and cell growth starts to accelerate to reach very high value, and at the end of this time point, actually the cell is lysing. So in a way, the cell is kind of thinning its cell wall and growing very fast, and it does not realize that the cell wall is becoming too thin over time, and then it undergoes a lysis by piercing the cell wall. Okay, and so this was very interesting to us because it suggested at least that there is some kind of form of mechanosensation potentially that will allow these cells actually to kind of get a sense of how the cell wall is being strained or stressed over time, and to kind of adapt during normal cell growth, the cell wall synthesis, in order to maintain some kind of homeostatic system that will allow them to remain alive. Okay, and so in the absence of this system, actually the cell wall will tend to undergo catastrophic event, to rupture and lead to cell death. Okay, but this begs a very fundamental question now, which is to try to understand how these components of the cell wall integrity pathway are actually able to probe the stress in the cell wall in the strain. And so, kind of classical question about mechanosensing. And so this is what I will discuss in the second part of my talk. So these are cell wall mechanosensors that I will talk about. And so this is a project that was driven by a postdoc, Ramakant Nelly Venkata, and Celia, an engineer in the lab. And so these are the two classical mechanosensors or sensors for the cell wall, and these are highly conserved. You find them in all fungi, basically, so from yeast to fungi. So there is 400 million years of evolution, and you find all these two type of sensors in all these fungi. And so there are two main families, actually. So there is the family of the whisk-type sensors and the family of the mid-type sensors. And in Pombe, actually, in fish and yeast, we only have two of these sensors, one that is called WISC1, which is of the whisk type, and MTL2, which is of the mid type. And so the WISC1 mutant undergoes a rate of lysis by piercing the cell wall at a rate of about 10 to 20%. MTL2 mutant also dies at a rate of 10 to 20% per cell cycle, and the Dalmel mutant is actually completely dead, suggesting that the two proteins are kind of supporting or complementing each other to support viability and integrity of the cell wall. Okay, and so the structure of this protein is very interesting. So these are single transmembrane protein. And so you start from a cytoplasmic tail, actually, that is very important probably to signal downstream in the cytoplasm and trigger the activation of the cell wall integrity pathway. And then you have two very large extracellular domains. The first one is called serine threonine rich domain, so the STR domain. It's highly manosylated, probably to allow it to maintain a rod shape. And then on top of this domain, there is another domain which is called the WISC domain, and also the CRD domain sometimes because it's very rich in cysteines. And the length of this sensor has been estimated to be actually very large, typically 40 to 50 nanometer in height. So they do not protrude out of the cell wall. They are within the cell wall, but they have these very, very long domains that are typically embedded within the cell wall matrix. Okay, and so the other sensors is called MTL2. It also has this STR domain, but it lacks this kind of WISC domain that is actually, so the WISC domain is a lectin-like domain that binds specifically polysaccharides, sugars in the cell wall. Okay, and so if you just simple experiment, and this was published actually by the group of Yolanda Sanchez many years ago, if you look at the localization of these sensors, WISC1 will tend to be more polarized, so it localizes mostly to cell tip, and MTL2 is more diffuse. It has this kind of broad pattern all around the cell. Okay, and so actually there have been a thought in the field that these sensors could act as bona fide mechanosensors, in part through this very important work that was performed by the lab of Yves Dufresne, where they are using AFM tips. And so what they did in this work actually is that they generated some chimera of this STR domain, and they were able to use an AFM in order to probe single molecules of these sensors, and they could show that actually if you apply a force, the sensor will deform linearly with force. So in a way, this is some kind of properties that will reflect some kind of mechanosensing properties. And so they named actually this sensor some kind of nanospring. And so people tend to call this WISC1 sensor or MTL2 sensor nanospring. And I just remind you that these sensors are upstream of the cell wall integrity pathway, so they perform a very important function, which is to monitor the integrity of the cell wall and to trigger some kind of adaptive cascade to ensure that the cell wall remain intact. Okay, but then the question that we had, and that was really missing in the field, is how these nanometric sensors are able to kind of probe the mechanics or the deformation of the cell wall at a scale that is much larger, at the correct, in a way, cellular scale. And the answer came from kind of a serendipitous observation, which it's always what happens in most labs actually, just by a simple experiment. So here what I would show are just a simple spinning disk time lapse where we filmed basically cells that are expressing WISC1 GFP and just growing onto each other in a standard agar pad. And we made the discovery that actually when the cells are dividing, right after the cell division, when the two hands are contacting each other, and I hope the movie will replay, but it doesn't matter, you have the time lapse here, you see that the two hands are going after each other after cell division, and you have this kind of bright cluster that forms exactly at the position where the two cells are contacting each other. And then later on, actually, because the cells are kind of contacting each other, they push onto each other and they slide past each other, so the contact kind of relaxes, and you see that the cluster is kind of vanishing or disappearing or dissolving. Okay, and this is not only twice the fluorescence of the plasma membrane. Actually, you can do some very simple quantification, and you see that the enrichment that you will get at the site where the two cells are contacting is about typically 10 to 15, depending on the situations. And so typically, you have a rising phase where the cluster will form, and then the cell will slide, rearrange, and then the cluster disappears. And so initially when we looked at that, we thought, well, this has to be something specific to cytokinesis or septation, because during cell division in walled cells, you have to rebuild the secondary septum, and so maybe there is something specific there, and that's why the WISC1 is kind of recruited massively to this site. But then by filming cells over and over, actually, so here these are kind of multiple time lapse that are stitched together in order for you to appreciate. We could see like all of these little clusters that were decorating all the sites where cells are contacting. So as soon as a cell is kind of contacting and pushing on the other one, you see that you have a tiny cluster that is kind of dynamic and tracking the site of cell contact. So I hope you can appreciate. Okay. So and so here you have a kind of a more stable image where you can appreciate that these clusters can form at any position around the cell. Basically, you just have to have two cells that grow onto each other. So this is relatively exciting because this is thought to be a mechanosensors. You have two cells that are growing onto each other that contact and probably kind of push onto their cell wall, and they cluster, or they really form this very bright enrichment in the WISC1 protein. And then I'll show you another physiological situation. So I've been talking about sexual reproduction in yeast. So this is how yeast cells actually mate. And when they mate, actually they will grow this kind of schmooze. So these are mating tips, and the cells are growing toward each other. Then they will kind of contact each other. And then the cell wall that is at the contact will become dissolved so that you can fuse the two cytoplasm and form a zygote. Okay, and during this process, so this is really a physiological process, you see that the cluster is actually heavily recruited exactly at the site where the two cells are kind of kissing each other, and you see these bright clusters. And actually the enrichment that you have during mating is a little bit enhanced as compared to during vegetative growth. Okay, and so this is kind of interesting, but in a way it makes it very difficult to kind of test mechanisms for the clustering and whether this is really mechanosensing, in part because if you just look at cells that are growing in agar pads, they touch each other, they may be exerting some forces, but then they slide past each other, so the clusters are only formed kind of transiently and they're not extremely bright. And so of course we thought about developing techniques to kind of force cells to grow onto each other. And so Armin Haupt, a former postdoc in the lab, had developed this relatively nice, very simple actually, microfluidic chambers, which are kind of optimized to grow cells in very large confinement, while they have access to a lot of nutrients. So in a way, the cells are very happy in terms of food in the channels, but they have very little space to grow away. Okay, and so you see that you can form kind of colonies, linear colonies, and the cells are forced to kind of grow onto each other, and you see that you have these very bright clusters that are decorating the site of contact between the two cells, and the enrichment is more stable in general. You see the clusters will stay more in time. And if you let, and these clusters now are formed at all tip-tip contact. You find them in 95% of the cells, so this is a really penetrant phenotype. And if you let the cells grow over and over, actually they kind of reorganize polarity. They really want to grow and to cover all the space. They push on the walls of the microchambers that you see here, or they push onto each other, and you see that now the these WISC1 clusters are kind of decorating all the interface, even the lateral contacts where the cell walls are pressing onto each other. Okay, and so the idea that we had actually is that these cells are very turgid, they grow onto each other, they push onto each other, and then as a result maybe the cell wall in between is kind of compressed or stressed, and so this is why this mechanosensor is being recruited there. And to test that, of course, you have to kind of be able to manipulate the mechanical stress in the cell wall, and so there is very simple way to do that in yeast, is that you can just expose them to very high osmolarity medium. So for instance, you can bath them in 1 molar sorbitol. This will tend to extrude the water from the cell and to reduce sugar pressure drastically. So you see that if you have these cells that are in channels, they form these multiple clusters at all sides of cell wall compression. You release pressure, and you see that within typically 10 to 20 minutes, you will lose most of the clusters. You have some that stay a little bit, and this is still a puzzle for me, that are very stable into the cell wall. And so you see the dynamic is relatively conserved, and there is a dose dependence. Basically, you have to apply some kind of sorbital or osmotic shock that is of sufficient magnitude to really remove all the stress. And so, of course, orbital shock can create some kind of problems or activate some specific pathways. So we also have developed many techniques just to manipulate stress dynamically. So here what we are doing, we grow cells into the channel, and then we come with a laser, and we can photoablate actually a couple of cells around a pair that is forming a clusters. And so because we're killing this cell, then basically the cell kind of deflates, like a crushed can basically, and it's being pushed by all the other cells. And so this allows these two daughter cells to kind of separate from each other and relax relax the tension at the contact between the two cell walls, and you see that again the cluster will vanish within typically 10 to 20 minutes. So this shows that you have upon mechanical stress into the cell wall that is created by Tjogor pressure and growth from from one cell onto another one, you have the rapid formation on a time scale of 10 to 15 minutes of these mechanosensor clusters, and this is reversible. So basically, they can also disappear very rapidly once the stress is released. Okay, and so of course you want to test if whether there is in a way more clusters that are being recruited as a function of mechanical stress, so you want to address kind of the dose dependence. And so here what we did is that we adapted the kind of the two-color segmentation system that we have in order to measure actually the cell wall thickness at the interface between two cells. And so for this, we grow pairs of cells, so some cells that are expressing the WISC1 sensor tagged with a GFP, and some cells that are just labeled for their plasma membrane with Psy1 mCherry. Okay, so this is an integral membrane protein that is just decorating the plasma membrane. Okay, and so if you just apply the same method that I introduced in the first part of my talk, you will see that if you trace a line here, you will be able to measure actually the sum of the thickness of the two opposed cell walls. Okay, and because WISC1 is actually a transmembrane protein, you can use WISC1 to detect the inner face of the cell wall in this cell. And what you see is that as cells are going onto each other, actually the thickness of the cell walls at the interface here is decreasing over time, and it can decrease up to 40 or 50%, so there is really a compression of the cell wall, a progressive compression that you can monitor in live, and at the same time you can see that the WISC1 GFP signal is kind of increasing over time, and there is an enrichment, a progressive enrichment that is kind of depending on the amount of compression. And so if you do this and repeat these experiments in many different cells or in different situations, you see that there is a dose dependence. So basically, the more the cell wall is being compressed, the more you recruit WISC1. Okay, so this shows and directly demonstrates that WISC1 is actually a bona fide cell wall mechanosensor. You press onto the cell wall and WISC1 gets recruited there and kind of senses that there is a problem with the cell wall. Okay, and so of course this is again a yeast system, and so we had to kind of identify some kind of the mechanisms that are responsible for driving WISC1 to the site of cell wall compression. And so for this, we built on our microchannel assays where you have clusters that are decorating everywhere, and we search for mutants that will affect this kind of process. And so one very big surprise is that actually this clustering phenotype is mostly independent of trafficking and polarity. And so this is very surprising because this is a membrane protein that is thought to be delivered by secretion and recycled by endocytosis. But at least at the time scale of the formation of these clusters, you do not need endocytosis or polarized secretion to form these clusters. So in a way, they can go by themselves. They don't need to be driven by polarity pathways or polarized secretion. And this is also a system that is completely independent of all the other components of the cell wall integrity pathway and other systems. And so it feels, by looking at this data and by making these screens relatively exhaustive, that the sensor can do it by itself. Basically, it can sense that there is a cell wall that is compressed and kind of accumulate there and rush there. And so to test the hypothesis that this could happen, we performed kind of FRAP experiments to monitor whether the sensor can diffuse or have different diffusive property around the cells, depending on the state of compression of the cell wall. And so for this, what we do is that we also mix we use this kind of two-color assay where we have cells that are expressing WISC1 GFP and other cells that are expressing just this passive membrane marker. And we exploit cells where the two cells are not compressing each other, and so where there is no clusters, and we can do some FRAP only in the green cells. And so what you see is that when you FRAP, there is a recovery that is mostly within the plasma membrane. Okay, so there is a lateral diffusion within the membrane that allows the signal to be recovered in the center. And this takes about, I will say this has a lifetime in free tips that are not clusters of about 180 seconds. So it's a very slow diffusive process. Within 3 minutes, basically, you will recover most of the fluorescence. And if you do the same experiment, but in cells where the cell wall is completely compressed, and when you have clusters, you see that diffusion is much hindered, actually. And so you have an enrichment, the mobility of the protein is now completely slowed down, and you have a dose dependence. You can extract some kind of diffusivity of WISC1 GFP as a function of the mechanical stress in the cell wall, and you see that you have typically, you're losing a factor 3 in terms of diffusivity. So this suggests, at least, this favors the mechanisms by which these clusters may accumulate, or the WISC1 protein may accumulate to the specific site where the cell wall is being compressed through some kind of local restricted lateral diffusivity. Okay, and so of course, then this begs the question of how, and so if the sensor can do it by itself, just by modulating its diffusive property, then you have to understand how the external domains of the sensor are interacting with the sellable, okay? And so for this, we generated some kind of truncation alleles where we can remove one by one, actually, each of the domains that are known to be important for the sensors. And so this is the full lens that I showed you. So you have these clusters that form everywhere. One first very interesting alleles is this one, where we can actually truncate the whole cytoplasmic tail. And so these sensors are actually probably incapable of communicating with the rest of the cell wall integrity pathways. So you really, kind of cut the link. And you see that even if they cannot, communicate with the rest of the cell, they are still able to form this cluster. So this is kind of supporting the idea that this clustering can form independently of the rest of the cell wall integrity pathway. But more importantly, what we found is that if you truncate the STR domain, so this long kind of nanospring domain, or this whisk domain, which is the kind of lectin line domain that is thought to bind to sellable polysaccharides, you see that you completely abolish the ability of the whisk1 sensor to detect sellable mechanical properties, or sellable stress, sorry, and to form these clusters. And so you have this quantification. So basically, you see that in these 3 alleles, so here this is just an allele where we remove the STR nanospring domain. Here this is an allele where we remove the whisk domain. And here this is an allele where we remove the full extracellular domain, the 2 extracellular domains. And you see that the enrichment is basically close to 2, which corresponds to the sum of the 2 opposed plasma membrane. Okay, but there is one caveat in this experiment, is that the whisk domain could actually be placed at a certain altitude within the cell wall. Again, I mentioned that these sensors are relatively large. They are 50 to 60, or they are thought to be 50 to 60 nanometer in length, and the cell wall itself is about 150 nanometer. And so maybe you need actually this domain to be placed at a certain altitude to be able to sense properly the mechanical stress in the cell wall. Okay, and so what we did actually is that we performed the experiment on the other sensor, which I did not discuss yet. And so the other sensor is called MTL2. And actually what we found is this sensor is incapable of making a very bright cluster. So you see that it does not accumulate at the site where the cell wall is compressed. We sometimes see this little accumulation away, and we don't really understand how it works. It's suggesting that at least MTL2 cannot probe local mechanical stress in the cell wall. But what we did is that now we took the STR domain from MTL2, and we kind of generated a chimera where we inserted this STR in place of the normal whisk domain. So basically, we do not have the normal STR of the whisk1 protein, but we have this MTL2 non-clustering STR domain, and then we add on top of it just this whisk domain. And you see here that you can recover or rescue the formation of this bright whisk1 cluster, suggesting that actually most of the ability for clustering at sites where the cell wall is compressed is encoded by this little domain, which is a lectin-like domain that interacts with polysaccharides. Okay, and so I will skip details, but basically based on these experiments and other that I did not have time to show, we have this hypothesis that basically when you apply forces onto the cell wall, then maybe you will restrict locally the diffusivity of the sensor, probably because you enhance the binding of this whisk domain with cell wall polysaccharides. And so we propose actually, in analogy with what has been done for integrins, that you have some kind of kinetic trap model that allows to form this very bright and to localize actually the mechanosensor to the site exactly where the force is applied and to where the cell wall is being stressed. And so, of course, this is reminiscent of mechanosensoring clustering in animal cells, especially integrins that will form also this kind of micrometric cluster in response to tensile force. But the difference here is that the cells are, of course, not exposed to kind of pulling force, but they are kind of exposed to compressive forces. And so this clustering actually has very important implication in terms of locally activating the cell wall integrity pathway and to ensure that survival and detection of mechanical stress around cells is being intact. Okay, and so in the last few minutes, I will come back to kind of the studies of Syllable Dynamics, and I will show you just like some kind of preliminary results. So this is a kind of ongoing project that is still not published or far from being published. This is a project that is driven by Louis Chevalier, PhD in the lab. And so here really the fascinating aspect that we're coming back is thinking about the process of tip growth is really the very large variation in terms of growth speed. And so I just wanted to try to align 3 movies that I will play exactly at the same rate just to give you a sense of the wide variety in terms of growth speed. So here the first movie, this is a Pombe cell, and you barely see it growing just because I had to play it so slow in order to see the other fungi grow. So these cells are growing up at about 0.03 micron per minute. This is Aspergillus nidulans. It's a widely studied model because it's in a way a cousin of Aspergillus fumigatus, which is a pathogen of humans, an important pathogen. And Aspergillus nidulans grows typically 10 times faster. So I hope you can appreciate that by the time the yeast cells has grown a little bit, the Aspergillus is growing. And this is a Neurospora crassa. This is kind of the champion of tip growth in fungi, one of the fastest growing cells that you can find on Earth. And if I play it, actually, I hope these things you see that it's actually going back and forth several times by the time Aspergillus has made a progress of 10 micron. Okay? And so this is fascinating because all these cells are, their polarized growth and their tip growth is regulated by similar processes, polarized secretion, cell wall assembly, and then cell wall deformation and turgor pressure. And so, we have to understand how you can achieve this kind of large variability in terms of tip growth, and especially how cell wall integrity can be adapted to these very rapid growing cells. Okay, and so for us, this is fun for morphogenesis. So what Louis is doing actually is that he's been developing some Syllable Dynamics analysis in very fast fungal cells. So I will replay this movie because this is a gorgeous movie actually. Oh, no, I hope it plays. No, don't. No, it doesn't play. We start again. No. Oh, too bad. Bon. No, nothing is playing. Well, I will play this one. So this one is less. Okay, so basically we've been developing the same cell wall thickness analysis in very rapid growing cells, and so I hope you could see actually the cell wall deforming very rapidly. And of course, these fungi are going so fast that you are able to capture a lot of information. You see little pieces of cell wall that can translate throughout the cell, and you see very dynamic, oscillation at cell tips and homeostatic system that maintain thickness. And so we've been using Aspergillus nidulans, and this is in collaboration with Miguel Peñalva in Spain, because he's an expert in the kind of the fungal genetics of this fungus. And so the other advance that Louis has been implementing is that we can not only now track the thickness around of the cell wall around living cells, but we can also couple that to the imaging of a structure that we call the Spitzenkorper in fungus. This is a structure that basically groups some kind of secretory vesicle, and that in a way represents a very good proxy for secretion and cell wall assembly. And so in a way, by making these movies and a lot of image analysis, we are able to compute from the kind of the mass transfer between cell wall synthesis and secretion, fabrication of new material, up to the deformation of the cell wall. And so in part because growth speed is very rapid, and you have phases of very rapid and slow phases of growth in this fungal hyphae, you see that you have oscillation. We can track at the same time how cell wall thickness will oscillate and how the Spitzenkorper is changing in order to modulate. And so these are kind of the things that we are exploring, and of course we are using mutants, etc. And so here I'm just showing you some events. These cells can also undergo lateral branching, so they form this kind of branch which represents some kind of de novo growth, and we can see polarity or polarized secretion that is forming, and cell wall that is kind of becoming thicker over time, and then eventually will become thinner, etc., etc. Okay, and so this is kind of the one fun project that we are developing. Another one that is even more crazy is that basically there are 400 million of years of evolution for fungi, and so we have a bunch of species. This is a great world if you work on morphogenesis, because you have a lot of different fungal hyphae that have different shapes, different growth speed, etc., and probably different cell walls as well. And so we've been kind of together with Flora Klinger-Schmidt, an engineer, and Louis Chevalier, the same PhD student, we've been kind of implementing our cell wall thickness measurement throughout the fungal kingdom. So of course we cannot do that for all the fungi, but we had to select some kind of classical models, and many of these models are genetically amenable, so we have to kind of find tricks to label the plasma membrane and to label the cell wall, etc. And I hope you can appreciate just by looking at this here. This is Candida albicans, a very important human fungal pathogen, or botrytis, an important plant pathogen. Neurospora is a plant pathogen. This is the classical baker yeast from which we are making the beer and the bread, Pombe and Aspergillus, and we have many more. Okay, and so we've been kind of measuring cell wall thickness, and so in addition to cell wall thickness, we can also measure kind of the material properties of the cell wall. And so for this, this is actually what Valeria had been published in 2019. We can combine cell wall thickness measurement with the deflation of the cell. Okay, so if you take a map of cell wall thickness and then you kind of pierce the cell wall, you will deflate the cell. Okay, and much like if you have a spring that is stretched, and if you relax it, you will know what is the kind of the elastic strain, okay? And you can compare this kind of deflation with different osmotic shock. And at the end, together with the thickness, the deflation, and the osmotic shock, you are able to compute not only the local cell wall thickness, but also the local cell wall Young elastic modulus that kind of reflect the material property, the composition, if you want, on the cell wall, and also kind of the sugar pressure. Okay, and so now that we have these tools, actually, we can ask, these are very rapidly growing cells, very large cells, and so I don't know how they're associated with a more rigid cell wall or a thinner cell wall or a higher sugar pressure, etc. And so the first thing that we found is that actually there is a very nice kind of so these are preliminary data, so I'm not sure that the conclusion will hold, but we see that there is a very nice kind of scaling for tip cell wall thickness, and so this shows that very rapidly growing cells actually are associated with a very thin cell walls, okay? The faster you grow, the thinner is the cell wall at cell tip, so that's quite interesting. If you look in terms of the cell wall bulk modulus, you see that you have some variation, but they are not as marked. Most of the, the cells have kind of a bulk modulus that will range between 30 and maybe 50 megapascal, and so this may potentially reflect that all these cell walls are composed about similar, I would say, building blocks, so chitin and glucan. So it could be that the material properties by themselves are mostly self-similar among these different species. And if you multiply actually these two quantities, you can calculate a surface modulus, which is kind of the effective stiffness of the cell wall. And you see that you have this remarkable scaling actually, where the cell wall stiffness is actually decreasing as the cells is growing faster. So this shows that very rapid cells are actually associated with a much softer cell wall at cell tip. And so this causes a problem, because if you're large, you grow very fast, you have a very soft cell wall, you may actually risk to lyse the cell wall very often. And so the most surprising effect that we found is that when we've been measuring turgor pressure, is that actually these rapidly growing cells are also associated with a much deflated cytoplasm. So rather the intuition was that if you had a cell that was growing fast, maybe the pressure inside the cell will be faster, but it's actually the opposite, and probably or potentially to safeguard integrity, the strategy for growing very rapidly, at least among fungal aphae, is to make a cell wall that is relatively soft and a pressure that is relatively low. And so this begs very interesting question about turgor pressure regulation as a function of cell size and cell growth, and the maintenance actually of cytoplasm properties. And so I think I've been talking quite enough. And so with that, I will acknowledge important people. So I mentioned quite a lot of work has been developed by Valeria Davi, a former PhD student in the lab. I also showed data from Daria Bonazzi and Armin Haupt, who are former PhD and postdoc, and Henri de Belli, who also participated in the establishment of the cell wall thickness measurement. And most of the image analysis were done by Hiro Tanimoto and Dmitry Ershov. The cell wall mechanosensor work is done by Celia in the lab and by Ramakant. And we have very important collaborations with Yolanda Sanchez, Miguel Peñalva, and also Areski Boudaoud, where we are developing basically models for mechanosensing, for instance, and also a lot of help from the imaging facility at Jacques Monod. And of course, I would like to thank the funding and you for your attention. Thank you.
Thank you very much for your talk. We have 10 minutes for questions.
Hi, thank you for a nice talk. I'm curious about why this force sensor would not be diluted by exocytosis, for example, or if there's a balance between endo and exo, or the viscosity of sugar somehow keep them together.
So that's a good question. So actually, if you have a mutant in endocytosis, what you see is that I think I showed you at the beginning, but maybe the sensor is mostly polarized, so it's predominantly localized at cell tip initially. A mutant in endocytosis, of course, recycling is defective, so you will tend to have the cluster that kind of decorates the whole cell membrane, so it's not polarized anymore, but yet, even in the absence of endocytosis, when you compress the cell wall, you modulate diffusivity of the sensor, and progressively you accumulate the sensor at the site of cell wall compression. Yes?
So it implies there's a balance between exocytosis, right?
It does exocytosis. Yes,
It's
there
like
is
the one
exocytosis and
shown
endocytosis.
recently with this membrane distribution of CD42 being accumulated. Is it similar, you think?
Yes, so there is exocytosis and endocytosis. You
Yeah.
need to pre-position kind of the sensor at the plasma membrane, but this is not required at least to form a sensor and to detect cell wall stress. You understand?
Yeah, yeah. Thank you.
Nico, so I was surprised in your experiments where you have a huge cell-cell contact. It seems that you never run out of the sensor. So either the sensor is here in excess or you have some sort of mechanoregulation of the expression. Did you look at that?
Yes, that's a good question. Maybe there is one little caveat in this experiment is that here we are looking at a kind of a time shot that reflects the history of many cells that have been growing for hours and forming contact and pushing onto each other. So part of these clusters are formed de novo. You can see them visually, but some others are actually formed from prehistory and they remain there. So these are multiple cell cycles. So of course there has been progressive translation, etc. Either you just, all the sensors are accumulated there, or there is upregulation through the PMK1 actually transcriptional system. The only thing I can tell you is that we can shut down PMK1 and you still form clusters. So you don't need PMK1 to form clusters, but it's possible that you enhance or you maintain kind of the large pool of clusters in these situations through regulated transcription.
Regarding the last part with the potential coupling between the trigger pressure, the thickness of the wall and the speed, could you try to switch some of your yeast that are slow growing into fast growing by modifying the pressure and looking at the size of the walls?
Yeah, so we've been working on yeast like back in the days. So that's very easy to decrease pressure and to show that the cells will grow slower and slower. And this you can do in all these fungus, and it will work. So if you decrease pressure, the cells will always systematically grow slower.
But the reverse?
But doing the opposite is like very difficult. So we've been trying to overexpress, for instance, regulator of turgor, but this kind of never works actually. So you cannot inflate much more the cell that it is because it may actually lies the cell wall.
Any other questions?
So so it was very nice. Thank you so much. So I had a question about this exocytosis vesicles when you see vesicular transport. So I was wondering, in your model, when you have these very fast growing cells, do you see more vesicles? I mean, did you try to correlate this?
Yeah. So that's a great question. So kind of the amount or the concentration of vesicles here will kind of give you a sense of kind of the remodeling rate or how much vesicles you're transporting. So this is exactly what we are trying to do. In general, yes, the fastest growing cells that also have kind of a larger pool of exocytic vesicles. And so this was known for many years, but it's not always the case. So for instance, you can have larger cells that have kind of a more diffuse cap here, and they will still grow faster. So it's kind of a mix between the rate at which you remodel the cell wall, which is encoded probably by polarized secretion, and also the kind of the mechanics and turgor pressure. Yeah.
We still have 5 minutes for questions if anyone is interested. I actually have a very super naive question about the design of your assay to measure cell wall thickness. You label the outer wall with lectins, which are proteins. Could you have labeled sugars, or would it have been interesting to directly label sugars?
So you cannot genetically encode tag the sugars. At least you cannot make the protein express a certain GFP that will bind to sugar. So that's why we have to add the lectin directly in the
But would we have the chemistry to functionalize the sugar if you wanted to access from the outside?
But the problem, you could do that, but then you will label like the sugar at a certain time, and then the cell will grow, and actually it's only the pre-labeled sugars that will inherit the labeling, and so I'm not sure this will work, but yeah, why not?
So, if we